Okay, so we are back with a discussion of the Human Genome Project, and this is where I get to be a science nerd. And we'll throw in some fun facts. It is enough for you to know what the Human Genome Project is. So I will explain that and how it makes a difference in your life as a nurse. And I will explain that also. In 1990, an international alliance of scientists attempted to identify every single base along the entire human genome and determine the actual number of genes. Genes are made up of these bases. They're little discrete units of information. If you think of a beaded necklace, each gene is, a bead. The string is the chromosome. And anything that makes up that bead, if it's little clay molecules or whatever, that's your bases. There are 3 billion bases in human DNA, and they only make sense if they're in exact order. Now, I don't know if you can recall, but the very first week, I think I told you that if you are looking at the person next to you, having a conversation, and the thought occurs to you, I have nothing in common with this person, you are 99.9% wrong. But even there, if we have 3 billion bases, determining the ones that are responsible for all of the variation in the people you see, that's still 3 million bases. A lot of little guanine, adenine, cytosine, thymine. And they have to be in a precise order in the right place on the right chromosome. So what they did was they took 24 healthy donors. They scraped their cheeks. This was in like Buffalo, New York. And they cut the DNA into pieces because their equipment could only read about 1,000 bases at a time. So you start with 3 billion bases on this long DNA. If you want more trivia facts, if you were to stretch out all the chromosomal material in a single cell in your body, it would stretch out to 6 feet. So imagine the task in front of them. That's really what I'm trying to give you an idea of. None of that is testable material. It's trivial facts, but it gives you an idea of the scope of what they were getting themselves into. So they had to cut it into smaller pieces and read 1,000 bases at a time. They did it in overlapping pieces so they could fit the parts back together in the right sequence, like a giant jigsaw puzzle where the pieces fit together in only one way. Now, the problem with this, not only was it tedious and painstaking, but some sequences are very complex. Think of the number pi, where the decimals do not repeat and they go on into infinity. Well, the bases don't go on into infinity, but they do go on a very long time. So some of these sequences didn't repeat at all. And some of them were very repetitive, like thymine, 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 or adenine, guanine, adenine, guanine, adenine, guanine. And they would go on for a while. So it was trying to put a jigsaw puzzle together where either none of the pieces looked like they fit or all of them looked exactly alike. That is how big this project was. However, in 2003, way ahead of schedule and under budget, good to know, right? The team announced the sequencing of the human genome was complete. Hooray, there was celebration across the world. However, there were still about 400 gaps, sequences of DNA that were either too complex or too repetitive to be mapped with technology at the time. The scientists kind of considered those pieces of DNA to be quote unquote junk DNA. They weren't important in coding for proteins. They didn't constitute genes. They may or may not have been correct. But in March 31st of 2022, almost 20 years later, those gaps were finally filled in and all of the bases were decoded. So at this point in history, the Human Genome Project is finally really and truly complete. We have an entire map of the human genome. Anyway, scientists thought that there would be about 100,000 genes in the human genome because we're so complex and evolved. But it turns out we only have about 20 to 25,000 genes in our genome. Some species, This is, again, trivia, not testing on it, but you might want to know. Water fleas and rice actually have more. It turns out that there are species of ferns that have 1,260 chromosomes. We only have 46. So the number of genes or the number of chromosomes doesn't necessarily indicate the complexity of the life form. It's what it's the actual proteins they code for and how they're assembled. Anyway, that's all fun facts, right? Now we get to what it means for us as nurses. So by mapping the bases, you can compare human DNA and look for differences, right? So if we know that 99.9% of it is the same, we don't have to look at that 99.9% of it. We only have to look at those 3 million bases that are different among humans. Now, to make it even more narrow of a focus, you can study populations that are described as having a population bottleneck, where the variation in the gene pool is not as significant, either due to geographical factors like some tribe living on an isolated island, or you can look at populations that for social reasons don't tend to intermarry with other groups. So an example of this can be researchers study the difference between members of an Ashkenazi Jewish community who lived to be centenarians and people who did not. So they found a difference in just 2 bases in the sequence that was consistent between these 2 groups. That difference was found more often than not. The individuals who lived to be more than 100 had a cytosine in a certain region of their DNA at much greater rates than those who had cytosine thymine or thymine on that little tag. So it was like 2 nucleotide pairs. You had like the cytosine to guanine in one sequence. And the people who did not live to be 100 had differences in that DNA. So using technology like that, differences in those single nucleotide pairs can be used to identify the genes for many illnesses or many traits. Not always illnesses. I mean, we think of healthcare, but there are people looking at other traits as well. Traits like height or right-handedness or hair color. So the differences in SNPs can be used to identify genes and traits that they control. A lot easier to do this with single gene traits than with diseases that have complex polygenic causes or epigenetic causes. So cystic fibrosis, easy to isolate. You're looking for a single nucleotide pair. Diabetes could be controlled by hundreds of genes. So you might see a lot of variation in the genes, and it's harder to pick up that pattern. You have to be sort of like one of those code breakers in the CIA to pick it up. So the Human Genome Project led to publication of the human genome on the internet. This information was free to the public. They did not hoard it for themselves. They did not try to patent it. They were really interested in the greater good of people. And this is important because this was one of the first times in the scientific community that we decided to go with big science. If you've taken evidence-based practice as a course, or you've looked into evidence-based practice in any of your clinical courses, you'll note that a research question is usually fairly narrow. What is the impact of an educational intervention with staff nurses on falls risk in a long-term care facility with 200 participants? You try to narrow it down as much as you can because you can make that information available quickly and kind of reach statistical power, all those things that are important to people who do research. This was a question of big science. They didn't know where it would end. We just want to see all the bases on the whole human genome. And it was usually frowned on, but now it kind of leads the way for other scientists to do, or other groups of scientists, it's not a one-man deal, to do bigger projects without necessarily an end game in mind. There's increased testing for genetic disorders and inborn errors of metabolism. So using that example of cystic fibrosis, The test for cystic fibrosis, and you can still use it, but it used to be a sweat test. You would test for the amount of chloride in someone's perspiration, and based on what you found, you could diagnose cystic fibrosis. Well, the problem is you wait until the child is symptomatic before you do it. There's a chance that there's already been damage to all the structures that are involved in cystic fibrosis, right? So we can initiate treatment earlier if we can find those disorders. In fact, CF is one of the disorders that's now on the newborn screening. So we can intervene a lot earlier. We don't have to wait for sweat testing. If we want to know the status of a sibling with cystic fibrosis, who's got cystic fibrosis, we can test that person's genome and know whether they have it, as opposed to doing a sweat test, waiting for results, and then when it comes back negative, we still don't know if they're a carrier. We just know they don't have the disease. So these are some of the contributions the Human Genome Project made to health and wellness. There has been a creation of a catalog of mutations related to cancer. Note that I said related to cancer. When we talk about cancer, you will see that a mutation doesn't necessarily cause cancer. It increases risk. But we now have a catalog of those mutations. We can test for them and determine a person's risk for that cancer and give them options for prophylactic treatment. We have also developed cheaper methods of sampling and analyzing DNA to make these breakthroughs in medicine more accessible to people, make them more palatable to insurers, and even to, get people to buy 23andMe online at Amazon. So we now have cheaper and more efficient methods of sampling and analyzing DNA. It's also been used in police work. So we can identify victims earlier. We can identify perpetrators of crimes. We can rule people out as suspects. So those cheaper methods of sampling and analyzing DNA is another contribution that the Human Genome Project made. Development of therapeutics tailored to individual genomic factors. For example, the Philadelphia chromosome is an anomaly that involves a translocation of a small piece of a chromosome to another chromosome where it doesn't belong. Sometimes you can have the right gene, but if you put it in the wrong place, it's going to do something weird. And that is what happens with the Philadelphia chromosome. There's a medication called imatinib. There's another one. I think it starts with a D. But it specifically targets the proteins that are created when this Philadelphia chromosome translocation occurs. So that is something that's very exciting that would not have been possible without the Human Genome Project. We also have ways to test how people metabolize medications. If you watch the video with the cases, you saw how Gary used that testing to find out he was an ultra rapid metabolizer. Well, that has significance not just in psychotropic medication, but in other medications that are maybe metabolized on that same pathway as well. So we can really customize medicine to the individual. And as this technology evolves and our understanding of the human genome evolves, we'll really be able to get better outcomes for our patients. And that is the end game here. The other thing, and I read this, of questionable significance, the study of ancient hominids to understand mutations involved in human evolution. If that's your kind of thing, maybe minor in anthropology, go ahead and study that. There's some interesting articles on that. I can give you the links to them if you really want them, but you'll have to email me because I don't want to confuse anyone with too much information. Other fun facts about the Human Genome Project, for those of you who are interested, is that for every $1 the U.S. government spent on the Human Genome Project, $141 was returned in revenue to the gross domestic product, the GDP, the amount of money that's available to the federal government. So it was not as costly as they thought. They actually came in under budget as smart people were turned on by this. They developed less costly methods of analyzing the DNA. So this was actually beneficial to the US economy. It was not just a giant waste of money, which is what people thought it was initially. Okay, so if you go on the National Institutes for Health, National Human Genome Research Institute's website, you will find that they are not done with the human genome. They are using it for cancer research. It's one of the biggest priorities. There's a whole branch just dedicated to cancer. Epigenetics, which is the study of how the environment plays with our genes, switches them on, switches them off, alters them to create different health conditions. Behavioral health and social health, um, how these things are impacted by the human genome. There is a branch of research that is dedicated to that. Population health. This is a big one because, ethnicity, race, a lot of times these terms are self identified. Um, so it makes it a little more difficult to study populations based on risk that is related to ethnicity. However, looking at the human genome, we can see certain haplotypes or like little groups of bases that are associated with different um, ancestral backgrounds. It's not a perfect science, but they can sort of see like, okay, where is our risk? What does this mean for, let's say, the Mediterranean, people of Mediterranean ancestry, people of Asian ancestry, people who come from this region of India as opposed to that region of India. So that's a big branch of research. Small molecule therapies for genetic disorders. I could not speak intelligently to that. I do know that there is a branch of research for that, and I hope to learn more. There's a branch of research for complex polygenic disorders. They're studying rare genetic disorders and inborn errors of metabolism. Even though these things affect fewer people, they are devastating, and we can maybe get better outcomes. Things like even PKU, phenylketonuria, is considered rare. But when it happens, it can cause mental retardation, or I'm sorry, intellectual disability is the correct term, unless the diet is altered. But if we alter the diet, condition does not become a problem. So there's a whole branch that studies the human genome with this. De novo mutations and their role in disease. That was, when we talked about the cases last week with Marfan syndrome, 25% of those cases are caused by de novo mutations. So understanding which sequence of bases is involved or which base pair and how that might happen and how different de novo mutations affect our health. Some of them are probably not clinically significant, and some of them might be very clinically significant. The role of the microbiome. This is big, big research, and genetic variants of microbial organisms. So we're not just studying our own genome, we're studying the genome of single cell organisms to understand how they affect our health. So those are all important things. I'm going to take a break here and we're going to return for my discussion of CRISPR. I do not have a complete or thorough understanding, but I have learned an awful lot and I would love to share it with you guys. It will become the basis for a lot of discussion on ethics and legal issues. So let's go ahead and pause here.